All right, so I was kind of interrupted there. Sorry, I can only do 15-minute videos, so it kind of stopped me on there. So we were talking about this dendritic cell with the hot dog bun, the MHC2, with the antigen or the hot dog in it. So this is all connected to our APC or antigen presenting cell, which was a dendritic cell. Then we have our helper T cell in green. So this is the big T cell here, and it has a specific TCR or T cell receptor. And we see that that binding, all right, that T cell receptor has a unique shape that mimics the shape of the antigen, and it's able to bind to it. You also notice that our T cell or helper Ts, we call them CD4 cells. So here's that CD4 molecule, and you can see it's kind of making a little attachment to our hot dog bun or MHC2 on our antigen presenting cell. So that's kind of like another step in there that's helping to um, make sure that we don't accidentally activate some cells when they shouldn't be activated. All right, so kind of a two-step recognition like we have now with logging into our D2L sites and emails. We have that two-factor authorization or authentication that has to happen. It's kind of that same thing here. We're double checking what's going on. All right, so let's move on to step 3, challenging with antigens. So B and T cells, when presented with antigens, will proliferate and differentiate. All right, so proliferation means we're making lots of them. So it says making the clones. So we're making just going through mitotic divisions and producing lots of them. So the lymphocytes make clones of themselves, and when in a couple of hours you got thousands of lymphocytes, all with that unique antigen receptor, right? Because that's the only thing we want to make a copy of, is that one that is able to bind to that specific antigen. And this growth is really quick. All right, it does happen pretty quickly. And so these new cells then start to crowd out a section of your lymph nodes. So why your lymph nodes swell is because you're making lots of cells, lots of lymphocytes. All right, so that's your proliferation right there. All right, and then differentiation, so they can become memory cells or they can become effector cells. So memory cells are going to remember that pathogen, all right? We've seen this guy before. Or the effector cells are the ones that are actually going to do the fighting. They're going to help to destroy those pathogens. So let's talk about these different effector cells, right? So we've got basically kind of two different types of cells, our memory and our effector cells. So what can happen? All right. So one group might follow the cytokine trail to the site of the infection. All right. So this happens typically about a week after, right? And we talked about having our helper T cells. So our helper T cells, remember, don't do any of the fighting themselves, but they do help to boost the fighting ability of local cells. Oops, I think I skipped there we go. This is the slide we need to be on. Um, and they do this by releasing cytokines. So the cytokines can help to call in more reinforcements to that site, and that helps to increase inflammation. They can communicate with the macrophages, which is what they're showing in here, and they get the macrophages to start gobbling up all these pathogens. So they're kind of extending the lifespan of the macrophages, so that they can continue fighting these pathogens. So the helper Ts help to kind of motivate these macrophages and keep them in the battle. So besides eating the pathogens, the macrophages also can kind of consume a lot of the cell waste or damaged cells too. All right, so helper T cells kind of do that. They don't directly fight, but they help to activate a whole bunch of other types of cells. We have what are called cytotoxic T cells, or remember we said that these had the CD8 on there. Sometimes these are also called killer T cells. So cytotoxic T cells are kind of the siblings of the helper T cells, but they have a very different job because the cytotoxic T cells are the killer cells. All right. So as we said, we call them killer Ts as well. So they're very efficient at killing other types of cells, other pathogens. So these cytotoxic Ts have the same unique receptors. So they have been activated, right? Um, so just like a helper T, the cytotoxic T needs MHC molecules. So they actually use the MHC1 molecules, which we said were kind of like a window on the cell. So we said these are unique and they help cells to identify one type of cell from another. So if we think of them as a window into the cell, um, we can we can see if a cell has been corrupted on the inside. All right, so we'll see if a cell has been infected. And so therefore we can identify whether that cell needs to be killed or not. And especially with viruses, right? Viruses get inside of a cell, they infect the cell, and then they burst out of the cell, and then the pathogen then starts to spread. So we kind of need to see what's inside of the cell. So your immune system has kind of developed a way that it can use these MHC1s to kind of tell what's going on inside of a cell and whether it's corrupted or not. So cytotoxic T cells use these MHC1s. So what will happen is cells will display different types of proteins on their MHC1s. And we said, once again, that's kind of unique to the cells. If it has a virus inside of that cell, it might be displaying a portion of the capsid of the virus. So it's kind of letting other parts of our immune system know that it's infected with a virus. So remember, MHC1s are only found on every nucleated cell of the body, and our cells are constantly breaking down proteins and recycling and reusing them. So then when the recycling happens, your cell kind of picks a random selection of pieces and transports them to the membrane to display on their surface with these MHC1s. So you kind of think of these MHC1s as kind of a showcase of the proteins that have been inside of the cell, right? That are part of the cell. So it's kind of like a fancy display window to showcase a selection of items. And there are many thousands of these display windows on the surface, and each one kind of gets refreshed with new proteins about once a day. So if it gets infected, it can easily kind of display what has been infected it. So dendritic cells will also post antigens they have collected into their MHC1s. And so that's kind of what we're seeing here. We see the helper T with the hot dog in the bun, so our MHC2. But here we also see the MHC1, that window onto the cell that the dendritic cell has, and it's making contact with our killer T or cytotoxic T cell. Okay, so dendritic cells can activate both helper Ts and cytotoxic Ts. Once the cytotoxic T has been activated, right? So the dendritic cell is saying, hey, this is what's out there. The cytotoxic T becomes activated, right? We know it's the right cytotoxic T to activate because of that binding that has occurred with the surface receptors. So the cytotoxic T then matches that antigen from the dendritic cell and it starts looking for it. And once it binds to something that has the same receptor, it releases chemicals, and those chemicals create a hole in the cell membrane of the infected cell. And then it produces granzymes that are also released into the cell, and it causes the cell to basically commit suicide or apoptosis. All right. So very deadly. All right. So it's secreting material into a cell that it knows is infected, right? So this guy then goes out searching for that specific MHC class 1. And if a cell's infected, it may be displaying that and it will bind to it and then cause it to die. All right. So cytotoxic Cytotoxic T cells are active in graft and organ rejection, once again, because of that MHC1 that it's using to identify the cells, right? We said those MHC1s are kind of unique from one person to the next. And so you want to make sure, um, you know, that it's able to recognize what is unique to you, right? Since they're different from person to person, um, this is a place where your immune system can recognize that a tissue or an organ that has been donated to you, if you've had a skin graft or you've had an organ, all right, a heart transplant, right, a liver transplant that has been donated to you. Um, Once that other organ is inside you, your immune system will then start to attack it and kill the organ. So this is why when you receive donated organs or grafts from other individuals, you usually take very strong medications to suppress your immune system basically for the rest of your life. Because it minimizes the chance that your immune systems will find foreign MHC class 1 molecules and then try and destroy those cells. All right. One last one that we want to talk about is our natural killer T, so other effector cells. So these guys are not activated by helper T cells, and they are a part of your innate immune response. So even though we've been talking about adaptive immune response, we're kind of throwing these guys in here. So they're part of the innate response, and they don't have to be activated by helper Ts. So natural killer cells do not look inside cells at all. They simply check to make sure that an MHC1 molecule is present. So it's an all or nothing type deal. So the reason they do this is they help to protect you from viruses and cancer. So they're the first ones to kind of attack this, right? So something that's foreign. So basically they're looking for that MHC1 to see if it's you or not. And if they find that the MHC1 is missing, then they're going to attack it. Okay? So the reason for this is that many viruses will force cells to stop making MHC1 molecules. So cytotoxic T cells then have a problem because then they can't see inside the cell anymore. They don't have that window inside that has the antigen of the virus. So they can't see if it's been infected. Whereas natural killer cells simply look to see if an MHC1 is present or not. So if it's present, good. If it's not present, it's going to kill the cell. Okay, so it's going to cause apoptosis. And so if the virus is inside, it traps the virus inside and kills it. All right. So as it says, cancer and usually viral infected cells lack MHC1 receptors. And when it's lacking, then your natural killer cells are going to come in and kill those cells. So one exception to this is malaria, which is kind of interesting. So malaria is caused by Plasmodium, which infects red blood cells. And what do we know about red blood cells? They don't have a nucleus. And if they don't have a nucleus, they don't have an MHC1 receptor. So they have no display window. So your cytotoxic T cells are not going to find the malaria inside of it at all. And your natural killer cells have come to recognize that red blood cells don't have that MHC1, so they're kind of the exception to the rule there. And they're not going to bother with them either. And then the Plasmodium kind of hangs out and survives. All right. So we said that differentiation was we can produce effector cells. So we have our helper Ts that are going to go out. We have our natural killer cells. We have our cytotoxic Ts, or they can produce memory cells. All right. So once again, whenever you hear that you're immune to a disease, it means you possess memory cells. It means that you have a living memory cell that remembers that specific enemy. And so if that enemy comes back, these cells stick around. They're kind of like your guardians to guard you. And they help to recognize that pathogen much more quickly than it did the first time around, right? So we don't have to make a long trip with our dendritic cell and do this whole recognition system again, because the memory cells can immediately activate and start producing effector cells.